PubMed articles on intestinal permeability. This slide was from 2017. When we made the slide, it was 14,883 articles in PubMed on intestinal permeability. I checked 2 hours ago. It was 14,930. That meant 50 more articles on intestinal permeability have been published in the last 2 weeks. That's how much emphasis is now on intestinal permeability as the gateway into the development of autoimmune diseases. We all should become experts on intestinal permeability in clinical practice. Not to teach and educate on, but really understanding how to look for it, how to treat it, what the obstacles are. And you'll get a lot of that. Some of the treatment from Dr. Salt after the break, and a lot more in the digestive module. They talk a lot about permeability in the digestive module. Today is about giving you the big picture overview. Increased epithelial permeability for antigens is a crucial primary or secondary event in the pathogenesis of several disorders. Could they say it any clearer? The title of this article was Gatekeeper Function of the Intestinal Epithelium. So, Mrs. Patient, when you eat foods, only the really small molecules can get through the cheesecloth into the bloodstream. The larger molecules have to keep going down the digestive tract, being broken down by the digestive enzymes until they're small enough to fit through the cheesecloth. It takes a whole lot longer to break down prime rib than it does a banana. That's one of the reasons why the digestive tract is 20, 25 feet long, is to give enough time to digest some of the foods that we eat. But when you've had inflammation in the intestines for quite a while and caused some damage to the intestines, and you may have a leaky gut, now these larger molecules can get right through into the bloodstream. And if that molecule right there, if that's chicken, your immune system in your bloodstream says, what's that macromolecule in the bloodstream? That's not something I can use to make new bone cells or brain cells. I better fight that. You make antibodies to chicken, or to tomatoes, or to basil, or to papaya. It doesn't matter what the food is. If it gets through a leaky gut before it's been broken down small enough, your immune system, trying to protect you, is going to fight that food. These are the people, you do a 90 food IgG panel, they come back sensitive to 20, 25 different foods. They say, oh my God, that's everything I eat. Well, of course it is. Your immune system's trying to protect you. You don't shut down the immune system. Rather, avoid the foods for a while, deal with the inflammation in the gut, allowing the epithelial lining to heal, to regenerate healthier cells without permeability, and then go back and check in 6 months. Everything's okay? Then, well, can I eat that food now? Well, I don't know. But go ahead and try again. We'll do it for a couple of months as long as you feel okay. And then we'll run the test one more time to make sure that you're okay. That's a rational way of approaching food-related disorders. So here is a drawing that demonstrates both transcellular permeability and paracellular permeability. Now, here's a pearl for you about transcellular permeability. It's called lipid raft transcytosis. Lipid, fats, raft, like a boat, transcytosis, right through the cell. Some of you may remember from anatomy and physiology classes, we absorb our fats right through the cells, not between the cells. But lipopolysaccharides will jump on the back of a fat molecule and get escorted right through the cell into the submucosa. LPS that gets into the bloodstream is called endotoxin. What's the end stage of accumulative endotoxin over the, oh, by the way, it doesn't stay in the submucosa. It gets into systemic circulation. You find it in the inguinal nodes, in the axillary nodes, in the spleen. You find it in the brain. You find antibodies to LPS in the brain, of course, in systemic circulation. And what's the end stage of years of endotoxin slowly leaching into the bloodstream because of a chronic permeability situation? What's the end stage of that? We call it sepsis. and it kills over 250,000 people a year in the U.S. My mother died from this. I know this one pretty well. And it goes on for years and years and years and years. That's why one of the biomarkers in testing for intestinal permeability is antibodies to LPS. Because lipopolysaccharides are not supposed to be in the bloodstream. And if you've got antibodies to lipopolysaccharides in the bloodstream, your immune system's trying to protect you. And if you've got elevated antibodies to LPS, how did the LPS get there? There's only a few ways it can get in. The most common way is pathogenic intestinal permeability. Now, intestinal permeability is transient. It occurs in everyone. That's how we absorb our nutrients, is through permeability of the intestines. But pathological intestinal permeability is when it's chronic, long-term, big gaps in between the cells, things like that. It was Professor Alessio Fasano, who now chairs the Department of Gastroenterology at Mass General at Harvard, and he spoke here a few years ago. Professor Fasano won the Linus Pauling Award here at IFM. He identified zonulin in the year 2000. We think he'll get the Nobel Prize someday. That usually takes 20 years, 25 years beforehand. But he identified zonulin as the gateway in the development of autoimmune diseases. This is a great article in Scientific American that came out in 2009. It's worth going to scientificamerican.com, I think it is, and look for this article, Surprises from Celiac Disease, and see how he writes about it. He's got an excellent way of expressing it. And he talks about, back then in 2009, he said it was a trilogy in the development of autoimmune diseases. And he used celiac as the example because it's the only one that's slam dunk, no arguments. We know for sure what the trigger is that sets it off. Wheat. But you have to have the gene. 93 to 97% of celiacs carry DQ2 or DQ8. A few don't, but the vast majority do. So you have to have the gene. Then you have to have leaky gut. And then you have a trigger. The trigger is wheat. You eat wheat. If you have intestinal permeability and you carry that gene, it will create celiac disease. But only 1%, 1 to 4%, depending on what country you go to or you look at, has celiac disease. But over 30% of the population carries DQ2 or DQ8. So it's not the gene. And everybody eats wheat, so it's not the environmental trigger. So what is it that sets it off? Why people get celiac disease, called loss of oral tolerance. And they talk about that in the digestive module, the weekend digestive module, why only a small percentage of people with the gene actually get the disease. But we have to understand the mechanism. So in 2009, it was a trio of causes. You had to have the gene, you had to have the environmental trigger, and you had to have a leaky gut. And so what they said back then was you can arrest the development of autoimmune diseases by healing the gut. Not that you reverse it all the time, but you arrest, meaning you stop the progression of the autoimmune disease by dealing with the gut. So look at this guy. What kind of a life has he lived? Vibrant, dynamic, full of piss and vinegar? I don't think so. Look at his slumped shoulders. You think he feels kind of beat up by life with this severe psoriasis? What about the environmental trigger being taken out? 1 month on a gluten-free diet. 1 month. Because you took out the environmental trigger and you arrested the development. And in this case, it reversed the disease, put it into full remission. Sometimes that happens. Often that happens, but not always. But I thought this is such a classic example how the environmental trigger can have such a big difference. It can be a toxin. It can be a chemical. It can be a food. Whatever the environmental trigger is and your genetic vulnerabilities will determine how it's going to manifest in your body. But I thought this is such a great photo of what is possible sometimes. So now we know from Professor Fasano it's not a trilogy. It's actually 5 components. This is 10 years later now. There are 5 components. First, the genetic propensity. Second, the environmental trigger. altering the microbiome, triggering inflammation in the gut, which creates increased intestinal permeability and translocation of those macromolecules, LPS, food molecules, into the bloodstream. A systemic immune response trying to protect you, and initiation of the autoimmune spectrum. And that goes on for years, damaging tissue, damaging tissue, damaging tissue, until there's enough tissue damage that symptoms become obvious. The autoimmune process can be arrested if the interaction between genes and environmental triggers is prevented by reestablishing intestinal barrier function. So this is not a talk on autoimmunity, this is a talk on the gut. The gut is the gateway in the development of autoimmune diseases. Right? That's why it's so important to understand this. And the possible causes of impairment of the intestinal barrier, there are many. You can see them here. There are many. Surprise for many people is that emotional stress can cause intestinal permeability. That when you produce stress hormones, they can cause intestinal permeability. Another surprise for many is when you learn that if you have the biomarkers of intestinal permeability, those are also some of the biomarkers of blood-brain barrier permeability. You get a leaky brain. You have a leaky gut, you likely have a leaky brain. How many people raised their hand just a few minutes ago about, yeah, it seems to affect my brain? Right? Yeah. Yeah. So the triggers of increased intestinal permeability, these are some of the triggers of increased intestinal permeability. The mechanism is the same in terms of it contributes to altering intestinal function and developing intestinal permeability. At which point, now you get the macromolecular antigens getting in, you get malabsorption, malnutrition, increased uptake of toxins and lipopolysaccharides. And it doesn't matter if it's coming from stress or if it's coming from strep. That probably is a good title for a paper, stress or strep. Take your pick, right? It doesn't matter. The mechanism is very similar. That's why we focus on the gut. So we're going to take a couple of minutes now at your tables and ask the question, what are the triggers and mediators in Joan's case? What are the most important, and then in your patient population, what are some of the most important triggers and mediators that you might look at? Okay, here we go. Next one is the gut microbiota. If you thought that there were a lot of studies on intestinal permeability, they dwarf the number of studies coming out on the intestinal microbiota. It is 100 trillion friends you may not know that you have. Proteins and metabolites generated by microbiome permeate the body. Of the small molecules found in healthy human blood, 36% are created by the gut microbiome. What? A third of everything in my bloodstream comes from the microbiota in my gut? Yes. Not from the food you've eaten. It's the exhaust of the microbiota. We call it the metabolome. But it's a third of the small molecules in your bloodstream at any one time. And there are so many different diseases now associated with an imbalance in the gut microbiota. I don't know of a disease that's not associated with it. I don't know of one. Imagine the scenario. A scientist says he's discovered a new organ in the body, comparable to the immune system in as much as it's made up of a collection of different types of cells, contains 150 times more genes than the host, it's host-specific, contains heritable components, modified by the diet, surgery, or antibiotics, and in its absence, nearly all aspects of host physiology are affected. All aspects of host physiology. That's your microbiome. With clear effects on physiologic, immunologic, metabolic processes in human health, aberrations in the gut microbiome and intestinal homeostasis have the capacity for multi-system effects. For most of my friends in functional medicine, if there were only one system they're going to treat, It's going to be the gut. And in treating the gut, the focus is on two things usually. That is, build a diverse microbiome and create an environment to reduce intestinal permeability. That is such a powerful 1-2 treatment approach. Irrespective of their presenting complaints, unless their presenting complaints are trauma, then it's different. Just as loss of honeybees from orchards or addition of an invasive species to a lake creates significant harm for the surrounding biosphere, so too it appears that small shifts in our microbiome caused by today's unhealthy diets can reverberate through human health. You change your microbiome in 24 hours by what you eat. It doesn't take a long time. Want to run an experiment? Have a couple of days of just lots of vegetables, plates of vegetables, a plate of vegetables at every meal. Cooked, doesn't have to be raw, but at every meal. And notice how you feel in 2 days if every meal, including breakfast, has a lot of varied vegetables, not just carrots. Don't eat a bag of carrots. Dr. Deanna Minnick, who's faculty here at IFM, she talks about the rainbow diet. Eat the rainbow diet. Lots of colors. And notice how you feel in a couple of days. It makes a difference. The mammalian immune system seems to be designed to control microorganisms, but in fact, it's controlled by microorganisms. And the impact factors on the microbiome, including genetics, lifestyle, early colonization, babies that were born natural childbirth or induced C-sections, medical practices of antibiotics, the hygiene theory, all of these things have an impact. You'll learn all about this as you start to dive into what impacts on the microbiome. I just want to bring up the point of how important the microbiome is. Critically important to all function in the body. So this tells us very similarly to what we saw earlier about permeability, but it's dysbiosis that is the middle link there that you didn't see in the earlier version of this slide. That all of these triggers create dysbiosis, which creates the inflammatory environment in the intestinal lumen, which allows intestinal permeability to occur. The microbiome is the missing link. When we talk about loss of tolerance, I talked earlier about celiac, loss of tolerance. It's the microbiome that's the determining factor of when you cross the line of tolerance. That's why it's so important. And our microbiota changes throughout our lifetime. The diversity changes. And there's lots of studies, they take animals and they take the microbiota of young animals and put it in the old ones. The old ones start functioning younger. And they take the microbiota of the old animals, put in the young ones start having more disease. You've all heard about fecal transplants. I'm not talking pros or cons, but that it impacts on the function of the receiver when you change the microbiota. So learn how to change the microbiota in a healthy, non-traumatic way. And this drawing, we think of salmonella as being a pathogen, but there is non-pathogenic salmonella in our microbiome. And what's its purpose? One of its purposes is it blocks ubiquination. What does that mean? NF-kappa B has a muzzle on it called inhibitor kappa B, kinase. And non-pathogenic salmonella keeps the muzzle on NF-kappa B. When you don't have enough non-pathogenic salmonella, the muzzle comes off and you activate more NF-kappa B action in the gut, more inflammation in the gut. So the metabolic activities of the microbiota, they ferment non-digestible residue, releasing short-chain fatty acids and vitamin K. They have trophic activities to help with building new cells. Mrs. Patient, you have an entire new body every 7 years. Every cell in your body regenerates, every cell. Some cells are really quick, like the inside lining of your guts every 2 to 3 days. Some cells are really slow. Bone cells are slow, brain cells are slow, but every cell regenerates. So are you regenerating healthier cells or more disease cells, right? When you work on creating more diversity of your microbiome, you create a healthier environment in your gut. What we eat influences the population and metabolic activity of our microbiome. This is very true. So gut dysbiosis and inflammation, altered gut function, creates systemic LPS, pro-inflammatory cytokines, endotoxemia, altered blood-brain barrier function, gut-brain axis dysfunction, and cerebellar and hippocampus dysfunction. That's just from another paper. There are many different papers that talk about how an altered gut microbiome will alter function at the endpoint. Altered microbiome creates this, creates this, creates this, creates this, creates that, and they come in with cerebellar degeneration, and we want to treat their cerebellum. They've got gait ataxia. You have to treat the gut as a component of your protocols to change the inflammatory cascade that's fueling the end-stage manifestation, whatever your genetic vulnerability is. Does that make sense? So this book came out by Martin Blaser, talks about the overuse of antibiotics fueling the dramatic increase in conditions such as obesity and type 1 diabetes, inflammatory bowel disease, and many others. And they've proven that in test animals again and again and again. Atopic inflammatory autoimmune disease has been linked to gut microbiota dysbiosis. Some cases, significant associations have been established between these diseases and the intake of antibiotics during early life. The first slide we started with, when in doubt, treat the gut. The next slide could have been, when treating the gut, address the microbiome. That's the first place you start in the gut, is addressing the microbiome, because its systemic impact factor is higher than anything else. It's the number one in terms of impact factors on gut dysfunction, impact factors on systemic dysfunction. Address the microbiome. It's not the sure cure for everything, but it's a prerequisite to addressing systemic inflammation. So the effects of antibiotics in children, this is a nice example. The light gray line on the bottom are kids that had no antibiotics whatsoever and development of inflammatory bowel diseases. The blue line are kids that had 2 prescriptions for antibiotics and development of inflammatory bowel diseases. And the green line are kids that had more than 2 prescriptions for antibiotics and development of inflammatory bowel diseases. Antibiotics, 1 dose of antibiotics changes your microbiome. 1 aspirin changes the microbiome. When you start reading the studies, your jaw drops again and again and again. How is it possible to maintain a healthy microbiome given how we live today? That's a very valid question. And we need more emphasis on addressing our microbiome, feeding our microbiome in a healthy way every single day. It becomes a prerequisite. It's a 101. You want to be healthy? Put this much attention on your microbiome every day, whatever that means in terms of your food selections or supplements. So when we look at the imbalances we're describing, pathogenic microbes versus beneficial microbes, if there's more pathogens, you've got dysbiosis. It's most prominent in the digestive tract, but it can also occur on any exposed surface or mucous membrane, such as the skin, the vagina, lungs, nose, sinus, ears, nails, eyes. Dr. Liz Lipsky, who teaches in the digestive module, told me a while ago, she said, you know, Tom, when a woman has breast cancer, the microbiome of that breast is very different than the microbiome of the other breast. And I didn't know that, really. So many of our tissues have microbiome. The brain has a microbiome. It's not just in the gut. Mechnikov won the 1908 first Nobel Prize in medicine, the first one, talking about the bacteria in the gut and the microbiome and why yogurt was healthy for people. So 5,893 PubMed articles indexed on dysbiosis as of last month. So it's just one of those factors when you're doing a functional medicine practice, you always want to consider towards the top of the list. As soon as you address the symptomatic picture, of course you want your patient feeling better as quickly as possible, so that's usually the first thing we address, is what can I do for symptomatic relief? But then in terms of going upstream, usually one of the very first things on the list is, what's the state of her microbiome, his or her microbiome? It's not so much about the microbe as it is about the effect of that microbe on a susceptible host. It's about the relationship between host and microbiome. So we're not looking for classic infections when we're talking about dysbiosis. In one patient, it may present as dermatitis or psoriasis. The same microbial imbalance in another patient can present as peripheral neuropathies or inflammatory arthritises. Often what we find when working with autoimmune inflammatory patients, they're having a pathogenic inflammatory response to a non-pathogenic microbe. So here we've taken the functional medicine model and substituted the microbiome in the center of it. Look at the impact factor of the GI ecosystem. What's not included in there? And the answer is nothing in the human body. The microbiome impacts systemically on any and almost every tissue. I can't say every because I've not read about every single tissue, but it impacts substantially. Using what you've learned, describe in layman's terms what's dysbiosis, what causes it, and why it may be of issue.